Hello everyone. Welcome to today's webinar. This webinar is titled Separation and Characterization of Macromolecular and Supramolecular Structures by Light Scattering. During the presentation, we will hear from Emma Jagar. I will now hand it over to Emma to get us started.
Welcome to Wyatt's webinar. First, I'd like to thank the moderator for kind introduction. In today's webinar, I'd like to discuss with you characterization of complex macromolecular and supramolecular structures by using various separation techniques coupled to light scattering. The content of this webinar is divided into two parts. The first part of the webinar comprises size separation of large particles in emulsions and suspensions by using asymmetrical flow field flow fractionation technique, which will be in the following shortly termed as AF4. In this case, AF4 was coupled solely to static and dynamic light scattering detectors in order to determine the size and shape characteristics of various soft particles like large unilamellar vesicles, lipid droplets, and polystyrene latex particles. The second part of my presentation deals with identification and quantification of side products formed during the synthesis of polystyrene block isoprene Miktoarm star copolymers and conjugates of protein and polyethylene glycol. In the latter case, we studied also conjugate aggregation in two different buffer solutions. Here we used a combination of various separation and characterization techniques, among which the use of a multi-detection system coupled either to SEC or AF4 was essential. In the introduction part, I'd just like to highlight the advantages of AF4 over more conventionally used size exclusion chromatography. AF4 is distinguished by a large separation range, from few nanometers to about a micrometer in size. AF4 has great separation power, especially in a submicrometer range. There is no degradation due to shear forces. The separation can be carried out not only of macromolecules in solution, as is the case in SEC, but also particles in emulsions and suspensions. AF4 is flexible also for separation of inhomogeneous samples, and moreover, fractions can be collected at the channel outlet and further offline characterized, since AF4 is a non-destructive technique. Besides water, organic solvents of low to medium polarity can be used as the mobile phases. As any other technique, also AF4 has its own limitations. The lower size limit is determined by the pore of the membrane mounted on the bottom channel plate and by the maximum cross-flow rate through the membrane. The upper size limit is set by inversion of elution order by steric elution, and this upper size limit depends on particle shape as well as particle size. Besides separation, also detection of eluting species coming out either from SEC column or AF4 channel is important. Here is a schematic presentation of equipment we are using in our laboratory. Both separation techniques, SEC and AF4, are coupled to a multi-detection system that consists of successively connected detectors. These are UV detector as the first concentration detector, multi-angle light scattering with embedded dynamic light scattering module at one of the observation angle, and refractive index detector as an additional concentration detector. By such equipment, we can determine the various parameters typical for macromolecules and particles. Size characteristics of eluting species can be determined solely by the use of light scattering detectors. With static light scattering, we determine the radius of gyration from the angular dependence of scattered light without the need of any concentration detector or knowing refractive index increment of sample. With dynamic light scattering, we determined hydrodynamic radius by measuring the Brownian motion of particles as a function of time. If we want to extract the molecular weight moments, the use of at least one concentration detector is necessary, since the intensity of light scattering signal is proportional to the product of molecular weight and concentration. By such systems, we can determine also the molecular weight distribution of samples, which is described by the so-called parameter dispersity. Furthermore, from the form factor or by plotting a double logarithmic plot of radius of gyration versus molecular weight, we can get also information on conformation of macromolecules as well as particle shape. In special cases, we can determine also the chemical composition along the molecular weight distribution of the complex two-component systems like synthetic copolymers, protein conjugate, glycosylated proteins, and complexes of membrane protein and detergents are. The only prerequisite is that the response factors of both concentration detectors are sufficiently different for both constituents of the two-component system. Since the first part of this webinar focuses on size and shape characterization of soft particles, let us look at the difference between both radii. Radius of gyration, also more correctly called root mean square radius, is determined by static light scattering measurements from the slope of the line representing the angular dependence of scattered light. Physically, radius of gyration represents the average distance of each mass point in a molecule or particle from its center of gravity, and it is described by this equation. On the other hand, hydrodynamic radius is determined by dynamic light scattering by measuring the intensity of scattered light as a function of time. From thus obtained correlation function, we can directly extract information on diffusion coefficient, and diffusion coefficient is further correlated to the hydrodynamic radius by the so-called Stokes-Einstein equation. Thus determined hydrodynamic radius is actually a radius of a hard sphere that has the same diffusion coefficient as the particle under examination. From both radii, we can determine also the so-called form factor, the value of which is characteristic for the particular macromolecular conformation or particle shape. In the following, I'm going to show you a few successful examples where the use of Wyatt's equipment was essential. The first example deals with large unilamellar vesicles, which are spherical particles composed of phospholipid membrane that surrounds an aqueous core. Vesicles were studied in two different sizes and at each size at two different chemical composition of the membrane. The membrane consisted of sphingomyelin and cholesterol. In parallel, we investigated also a compact system of lipid droplets, which instead of flexible aqueous core consisted of hydrophobic lipids. The surface of lipid droplets was covered by sphingomyelin and cholesterol. Lipid droplets were studied in one size at two different chemical compositions. The performance of dynamic light scattering detector in flow mode was checked by measuring the suspensions of polystyrene latex particles of different but defined size and narrow size distributions, so they served us as the Nanosphere size standard. Our goal was on one side to estimate a range of flow velocities at which the particle sizing by dynamic light scattering is accurate. On the other side, we wish to find out if AF4 coupled to light scattering detectors can be used as a tool for monitoring vesicle deformation in flow conditions. Namely, it is known that large vesicles are, due to flexible aqueous core and bending elasticity of the membrane, prone to shape deformation under influence of the flow. To reach this goal, we analyzed vesicles by AF4 at three different detector flow rates. These were 0.2, 0.5, and 1 milliliter per minute. Please note that detector flow rate is actually a longitudinal flow passing the detectors in elution mode of AF4 experiments. Dynamic and static light scattering measurements were performed also in batch mode, where the samples were not subjected to flow conditions and were also not separated by size prior to measurements. So let us look how the applied longitudinal flow in AF4 affects the extracted radii from static and dynamic light scattering measurements. In the first set of experiments, we adjusted at each chosen detector flow rate the velocity of the crossflow, since we wish to elute the whole samples within operating crossflow in order to ensure fractionation of samples according to size. Elution profiles recorded at 90 degrees scattering angle under optimal experimental conditions are presented in these figures. At all detector flow rates, AF4 successfully separated lipid droplets and vesicles of all sizes and compositions according to hydrodynamic size, as indicated by continuously increasing radius of gyration as a function of elution time. From static and dynamic light scattering measurements, we determine the average radii at all three detector flow rates. Radius of gyration appears to be unaffected by a longitudinal flow, while hydrodynamic radius decreases with increasing detector flow rates. The highest reduction in hydrodynamic size with increasing flow rate was observed for the largest particles. The difference in determined hydrodynamic radius was about 60%. Such huge reduction in hydrodynamic radius could be an indication of vesicle deformation in flow. However, the reduction in hydrodynamic size was observed also for lipid droplets, although in this case to a much lesser extent. Additionally, for lipid droplets, we did not expect any deformation in flow since they consist of compact lipid core. Since these results were quite unexpected, we checked the performance of our instrument by measuring the polystyrene nanosphere size standards of known and defined size. The curves representing radius of gyration as a function of elution time are flat lines with no slopes, since standards have uniform size distribution. Similarly, as lipid droplets and vesicles, also nanosphere size standards show independent radii of gyration on the detector flow rate. On the other hand, hydrodynamic radii were underestimated at faster detector flow rates. The only exception is the standard of 30 nanometer in size, where both radii are found to be independent on flow conditions used. Based on these results, the reduction in hydrodynamic size in flow conditions was attributed to incorrect calculation of diffusion coefficient from the correlation function obtained by dynamic light scattering detector in flow mode, since our software does not differentiate the translational motion of particles from their Brownian motion. The radii determined in batch as well as in flow mode for all soft particles studied are collected in these figures. Regardless of the detector flow rate, the determined radii of gyration are for all soft particles comparable to the values determined in batch mode. The radii determined in batch mode are represented at zero detector flow rate. On the contrary, dynamic light scattering in flow mode gives accurate hydrodynamic radii only at the slowest longitudinal flow of 0.2 milliliter per minute. With increasing longitudinal flow velocity, the error in determination of hydrodynamic size rapidly increases. The error depends only on the detector flow rate and particle size, but not on the type of the particle studied. The only exceptions are 30 nanometer polystyrene latex standard, for which both radii were found to be unaffected by the flow conditions, and 250 nanometer polystyrene latex standard, for which we determined underestimated hydrodynamic radius already at the slowest detector flow rate. Due Due to incorrect determination of hydrodynamic radius at higher detector flow rates, the calculation of shape factor was reasonable only at the slowest detector flow rate. The shape factors determined at 0.2 milliliter per minute are comparable to those determined in batch mode. The values of shape factors for lipid droplets reveal homogeneous spherical structure, while the shape factors of vesicles indicate their spherical shell structure, which was also expected. Due to incorrect determination of hydrodynamic size at faster longitudinal flow rates, the shape factor cannot be used as a parameter from which we can infer on particle shape and thus on vesicle deformation. Therefore, we had to find another way how to extract the particle shape, in this case solely from static light scattering data recorded in flow mode. Namely, it is known that angular dependence of scattered light is shape dependent as long as the particles have uniform size distribution. This should also be the case at each elution time over the peaks in fractograms of polydispersed samples if the samples are successfully separated by size. For this reason, we constructed the so-called 3D plots, which represent the angular dependence of scattered light as a function of elution time. Light scattering patterns of small vesicles and lipid droplets are very similar to those of polystyrene size standards up to 100 nanometer in radius. In all these cases, the light scattering intensity continuously decreases with increasing observation angle in the whole range of elution times. The only difference between these soft particles is in the slope of the lines, which is due to their different sizes. Such light scattering patterns are typical for the particles much smaller in size from the laser wavelength, and their light scattering patterns can be described by Rayleigh-Debye scattering functions. On the contrary, angular dependence of scattered light of the largest vesicles significantly differs from small particles. At longer elution times, where large particles elute, the light scattering intensity rapidly decreases with increasing scattering angle up to the minimum, above which the light scattering intensity slightly increases up to a maximum and then again gradually decreases toward the largest angles of observation. Such angular dependence of scattered light is very similar to that observed for polystyrene latex standard of 250 nanometer in size and is typical for particles with sizes close to the laser wavelength. Such particles are classified as demi-scatterers. A difference in light scattering pattern between vesicles and the largest polystyrene latex standard is only in the position of the minima and maxima. Such differences are otherwise typical for spherical and spheroidal particles. Therefore, these results could be an indication of vesicles deformation. However, such differences can also arise from different internal structures of these particles and their different refractive indices. Therefore, based solely on the light scattering patterns, we were not able to make a reliable conclusion on vesicle deformation. Another way to extract the particle shape solely from static light scattering data is to plot the so-called dissymmetry factor as a function of particle size, since this curve depends on particle shape. Angular dissymmetry factor is the ratio of light scattering intensities at supplementary angles, in our case at 43 degrees. The dissymmetry factors were calculated for polystyrene latex standards and for large vesicles at each elution time over the peaks in their fractograms, which were recorded at the slowest and at the fastest longitudinal flows. At all investigated detector flow rates, the dissymmetry factor for vesicles initially increases in accordance with the dissymmetry factors of polystyrene latex standards. Above about 100 nanometer, the dissymmetry factor of vesicles increases with size even faster, up to a maximum above which it rapidly decreases. Such a shape of the curve is ascribed to the fact that vesicles before the maximum scattered light according to Rayleigh-Debye theory, while the vesicles above the maximum scattered light according to Me theory. Some differences in the symmetry factors were observed for the largest polystyrene latex standard and the vesicles of comparable size. However, such differences could also be due to their different internal structure and chemical composition. Since also the symmetry factor did not give us an ambiguous answer regarding vesicle deformation in flow, we presented static light scattering data in the form of Kratky plots. Kratky plots were constructed at different elution times in the fractograms of large vesicles, which were recorded at the slowest and at the fastest longitudinal flows. These figures also show the theoretical Kratky plots for the hollow sphere and the rod-like shape. Kratky plots of vesicles exhibit, irrespective of the elution time where the light scattering data were extracted, and irrespective of the flow rate, a bell-shaped profile typical for hollow sphere structure. Finally, these results reveal that our vesicles did not undergo deformation under the flow conditions used in this study. So let us look what we have learned from this presentation. Radius of gyration is independent of flow rate. This is also true for hydrodynamic radius for particles with sizes up to about 50 nanometer. For larger particles with sizes between 50 and 200 nanometer in size, hydrodynamic radius is correctly determined only at the slowest detector flow rate. At faster flow rates, hydrodynamic radius is underestimated. Therefore, the particle shape of large particles cannot be inferred from the shape factors determined at fast longitudinal flow rates. In such cases, particle shape can be extracted from static light scattering data presented in the form of Kratky plots. Our vesicles did not undergo deformation under the flow condition used in this study, as indicated their Kratky plots. Next example deals with polystyrene block isoprene Miktoarm star copolymers, which are considered as complex polymers since they can simultaneously show distributions in molecular weight, chemical composition, and chain architecture. Additionally, residual Homo polymers can be present in the samples. Here, our goal was to identify and quantify side products formed during their synthesis. For this purpose, we use a combination of various separation and characterization techniques. Miktoarm star copolymers were synthesized by anionic polymerization of individual Homo polymers and their subsequent coupling to the chlorsilane linking agents of different functionality. Theoretically, all samples consist of one long polystyrene and several number of polyisoprene arms, the length of which decreases with increasing number of arms in the star. So, all samples should have about the same molecular weight and chemical composition. First, Miktoarm stars were analyzed by size exclusion chromatography coupled to a multi-detection system. SEC chromatograms of all stars reveal three distinct features. All samples exhibit shoulders at high molecular weight ends of the distribution curves, which indicate the presence of high molecular weight species in the samples. Their amount was higher for stars containing higher number of arms. At larger elution volumes, SEC chromatograms show low-intensity peaks, which were detected only by the refractive index detector, but not by the UV detector. Since polyisoprene is invisible for the UV detector, these signals were ascribed to the presence of residual polyisoprene homopolymers in the samples. The curves representing the molecular weight as a function of elution volume show different slopes over the peaks in the chromatograms, which clearly indicate the presence of different constituents in the samples. most probably the stars containing different number of arms. However, the constituents were not sufficiently separated by size exclusion chromatography. This was not surprising since the hydrodynamic volume of stars changes only slightly with the alteration of number of arms in the star. For better separation of different constituents in Miktoarm star copolymers, we used liquid adsorption chromatography on a C18 reversed phased column. The separation in liquid adsorption chromatography is directed by the enthalpic interaction of the solutes with stationary phase. In the first set of experiments, we studied the elution behavior of polystyrene and polyisoprene homopolymers of different and defined molecular weight and narrow molecular weight distributions. Under the chosen chromatographic conditions, the polystyrene homopolymers elute in size exclusion mode, that is from higher to lower molecular weights. On the other hand, the polyisoprene homopolymers elute in adsorption mode, that is from lower to higher molecular weights. HPLC chromatograms of Miktoarm star copolymers reveal much larger number of peaks than they were found in their SEC traces. These results thus reveal higher resolution power of HPLC as compared to SEC. Note that the region at small elution volume is magnified and represents the presence of residual polystyrene in the samples. At higher elution volumes, the number of peaks depends on the type of Miktoarm stars. All samples show above and below these intense signals the peaks of significantly lower intensities, which were visible only after magnification of the chromatograms. Based on elution behavior of both homopolymers, we expected that the constituents of the stars elute in a sequence of increasing content of polyisoprene, which was also proved by collecting the fractions at the column outlet and their further analysis. In order to get better insight into the complex composition of stars, we coupled HPLC with size exclusion chromatography into a two-dimensional liquid chromatographic system in order to simultaneously determine the variation in composition and molecular weight. The constituents of Miktoarm star copolymers elute in the first dimension by adsorption mode in a sequence of increasing polyisoprene content. This dimension is presented in y-axis. In the second dimension, each fraction coming out from HPLC is eluted by size exclusion mode according to the hydrodynamic size. The number and shape of eluting spot in 2D plots depend on the type of Miktoarm star. 2D plots clearly reveal the presence of several constituents in the samples. However, exact identification of individual constituents coming out either from HPLC or 2D system by using only the concentration detectors is not possible. In order to exactly identify the constituents of the Miktoarm stars, the fractions coming out from HPLC were collected at the column outline. Then they were further offline characterized by a size exclusion chromatography coupled to a multi-detection system. Since UV detector responds solely to the polystyrene component, while the refractive index detects both polystyrene and polyisoprene, we were able to determine the molecular weights of both components of the chemically homogeneous fractions. These figures show the refractive index chromatograms of the most intensive fractions of the 3 and 4 arm stars, together with the molecular weights of polystyrene and polyisoprene components as a function of elution volume. The fractions that are presented in samples in trace amounts are not shown. For the 3-arm star, the calculated molecular weights of the polystyrene and polyisoprene components are comparable. By dividing these values with the molecular weights of the corresponding homopolymers which were used for this star synthesis, we were able to determine also the exact number of individual arms in this star and also the star architecture. In this way, we found out that the most intensive fraction of the 3-arm star is a desired product consisting of one long polystyrene and two shorter polyisoprene arms. For the two most intensive fractions of the four-arm star, the multi-detection system reveals the same molecular weight of polystyrene component, while the molecular weight of polyisoprene component was higher for later eluting fraction. Again, by knowing the molecular weights of both homopolymers, it was clear that the early eluting fraction is a side product, since it consists of one polyisoprene arm less than it is predicted from the theoretical structure of this star. The later eluting fraction is actually a desired product with a predicted structure that is one long polystyrene and three shorter polyisoprene arms. Similar results were obtained for the 6 and the 8-arm stars. Both samples consist mainly of stars containing lower number of arms than it was predicted from their theoretical structures. The desired products are present in these samples only in trace amounts. For these two samples, we detected also large amounts of high molar mass species which were formed either by coupling of more than one long polystyrene arm to the chlorsilane core or by coupling of two silane functional groups into the siloxane bond. Such stars contain a larger number of arms than it is predicted from the core functionality. The summary of this work reveals higher resolution power of liquid adsorption chromatography as compared to size exclusion chromatography for separation of star constituents. Two-dimensional liquid chromatography gives better insight into inhomogeneity of the samples. By analyzing the chemically homogeneous fractions with size exclusion chromatography coupled to a multi-detection system, we were able to determine exact chemical composition as well as architecture of individual star constituents. Last example involves characterization of pegylated protein conjugate. Pegylation is considered as one of the most commonly used and successful techniques so far developed to enlarge the hydrodynamic volume of proteins. In this way, the residence time of biopharmaceuticals in the treated organism is significantly extended, which results in improved therapeutic efficiency. Due to safety issues of biopharmaceutical drugs, their exact and detailed characterization is extremely important. Therefore, our goal in this case was to identify the presence of unreacted protein and/or polyethylene glycol in the conjugate sample, to determine the degree of protein pegylation, and to determine the amount of aggregates in different buffer solutions. Protein conjugate was stored in two buffer solutions. One of them contained sorbitol, and in this buffer solution, the conjugate concentration was about 5 times higher than in the buffer solution without sorbitol. The fractograms of conjugate sample in both storage solutions show 3 peaks. The most intensive one is due to the conjugate. At shorter elution time, we observed very low intensity peaks visible only after magnification of the chromatogram. This peak reveals the presence of unreacted protein. At longer elution times, we can see the presence of a small amount of aggregated species. It should be noted that for the same separation of aggregates from the conjugate peak by size exclusion chromatography, we had to use 2 SEC columns, which results in longer analysis time. In addition, the amount of detected aggregates was lower by size exclusion chromatography, which was ascribed to irreversible adsorption of aggregated species on the column packing material. The amount of aggregated species was found to be lower in buffer solution containing sorbitol, despite the conjugate concentration was much higher in this case. These results reveal that sorbitol is an efficient agent for preventing conjugate and/or protein aggregation. The chemical composition of the conjugate sample was determined by the use of a multi-detection system. In this case, only the protein component of the conjugate was UV sensitive, while the refractive index detector responds to both constituents of the conjugate. In this way, we determined not only the molecular weight of the conjugate, but also the molecular weights of both conjugate components. Since the molecular weights of the protein and polyethylene glycol components of the conjugate were in good agreement with the values determined for the pure protein and PEG reagent, we concluded that conjugate is a monopegylated. This means that only one polyethylene glycol chain is covalently bound to the protein molecule. Multi-top mass spectra of both conjugates undoubtedly revealed the presence of unreacted protein, while the aggregates were not detected by this technique. Other two signals in mass spectrum belong to the mass ion and the doubly charged ions of the protein conjugate. In the magnified mass spectra, we can see repeating unit of polyethylene glycol imposed on the conjugate peak. In conclusion, this work reveals that AF4 coupled to a multi-detection system is appropriate technique not only to study protein aggregation, but also to determine the degree of protein pegylation. Multi-top mass spectrometry is appropriate for identification of unreacted protein, but not for studying the aggregates. Overall, the message of this webinar is that dynamic light scattering detector underestimates hydrodynamic radius of large particles at higher detector flow rates. In such cases, the shape factor is inappropriate parameter for determination of particle shape. AF4 coupled solely to multi-angle light scattering detector is suitable for studying particle size distribution, as well as the shape of large enough particles if static light scattering data are presented in the form of Krutkey plots. We once again prove that AF4 coupled to light scattering detectors is a suitable method for studying aggregation in macromolecular solutions. In addition, multi-detection system coupled either to size exclusion chromatography or asymmetrical flow field flow fractionation technique allows us to determine exact chemical composition of chemically homogeneous samples. In the end, I would like to thank Wyatt for kind invitation and giving me a chance to present our work at this webinar. I would also like to acknowledge the Slovenian Ministry and Research Agency for funding and the research groups collaborating with us on the presented topics. Last but not least, I'd like to thank all of you for attending this webinar and for your kind attention.
Okay, Emma, would it be possible to characterize something copolymers based on the ratio of RG-RH as you described in the first part of your presentation?
You mean Miktoarm copolymers?
Miktoarm, yes, sorry, thank you.
Yeah, actually yes, because the shape factor for star copolymers is different than for a random coil conformation. For example, theoretical value of the shape factor for star architecture is 1.33 and that of random coil is 1.78. But unfortunately, in our case at that time, we didn't have DLS model embedded in the system.
Okay. Is there any formula for having a correction factor to adjust the flow rate effect in RH calculation via QELS?
Unfortunately not, because this factor should also depend on the angle at which the model is mounted. But in Wyatt's company works on that problem, and as far as I know, they they developed already a flow cell that extends the range of accurate DLS measurements of up to about 300 nanometer in size for a flow rate up to about 0.3 milliliter per minute.
Okay, thank you for that. Can we use Kratky plots for the structural analysis, even in the FFF-MALS system, or is it only possible in FFF-MALS system?
Unfortunately, it is only possible in FFF-MALS system because in SEC the radius are too low. In SEC by SEC you can analyze macromolecules or particles of up to 30 nanometer in size, and for such low particles you cannot draw a Kratky plot.
Okay.
You need particles which are about 100 nanometer or more.
I see. Okay, thank you. Can multi-detector conjugate analysis be carried out on large unlamellar vesicles or lipid droplets to determine if they are encapsulating genetic material, a protein, or a drug compound?
In theory, I think it can be used. However, in the case of large unilamellar vesicles and lipid droplets, usually the amount of the sample loaded in the channel is small, since these particles scatter light intensely. So we can get a nice late scattering signal, but the intensity of the refractive index detector response is usually too low, especially when small amounts of samples are available, which is the case for exosomes. Additionally, the contribution of low molecular weight drugs to the total light scattering intensity will be most probably too low. In our lab, we try to determine the amount of protein prepores and pores formed on artificial lipid droplets and large unilamellar vesicles by a multi-detection system. However, the intensity of the UV signal due to the protein component was too low, which means that other more sensitive concentration detectors should be used instead of the UV in order to monitor the small amount of the protein component. For example, fluorescence detectors. Nevertheless, the loading capacity of the low molecular weight drugs with high specific extinction coefficients, as well as proteins or DNA, can be evaluated by comparing a ratio of the intensities of the two concentration detectors if the signal responses of both concentration detectors are high enough and they also differ enough for both constituents. If only the drug is UV active at a chosen wavelength of the UV detector, its amount can be determined solely by the UV detector.
Okay. Are the methods that you talked about suitable for molecules of molecular weight below 10 K?
Sorry, can you repeat the question?
Yes. Are the methods that you talked about suitable for molecules of molecular weight below 10 K?
Below 10K, you mean multi-detection or multi-detection system? Below 10K, it is very low molecular weight.
Uh-huh.
I don't know. Actually, we didn't work with such samples, so it is hard to say. But nevertheless, the intensity of the signals of all three signals should be high enough, which is for 10K maybe questionable.
Okay. What information do you need to know about conjugates, such as pegylated proteins or Miktoarm copolymers, in order to determine their composition by multi-detector conjugate analysis?
For complex two-component systems, the best results are obtained if only one component of the two-component system is UV active at the chosen wavelength of the UV detector, while the other component is invisible at this wavelength. So for determination of chemical composition by a multi-detection system, we need to know the values of specific refractive index increments of both constituents as well as specific extinction coefficients of both constituents. It should also be noted that accurate determination of delay volumes between the detectors is also very important when multi-detection system is intended to be used.
Okay. Let me just remind the audience that if you would like to ask a question, please type your query into the Q&A box. I'm not seeing too many more questions, but we do have a few more in the queue, so please, we have a few more minutes. Let's see. In which cases would you prefer SEC over FFF for use with light scattering to characterize macromolecules? And when would you prefer FFF?
You mean only for macromolecules or also particles?
Macromolecules.
Okay. SEC is preferred for smaller size macromolecules where AF4 encounters issues with low sample mass recovery due to the passage of the sample through the membrane, or when low mass recovery is a consequence of absorption of the sample on the AF4 membrane. SEC is also invaluable for determination of molecular weight characteristics of polymers that are molecularly dissolved only in organic solvents of high polarity, like hexafluoroisopropanol, DMSO, DMF. Otherwise, FFF is distinguished by much larger separation range, so you can analyze macromolecules of very high molecular weights of few millions, since as we know there are only few SEC columns on the market available for separation of such high molecular weight species.
And that is all the time we have. Thank you again, Emma, for your fascinating presentation.